Hello biologists, welcome to today's S-Biology Daily Revision Task. Right, in today's revision task, you're asked to produce a detailed diagram of the fluid mosaic model of a membrane structure, explaining its roles and its components. Right, this is a crucial skill that you've got to develop, and it's a crucial diagram that you need to know and understand, because lots of other things build upon it, especially when you get into A2 and you start looking at neuronal membranes and such forth. It'll be quite often talked about in exam questions where they'll ask you about certain features and how certain features interact and its functions. Hello and welcome to this A-level biology revision video on cell membranes. The basis of all cell membranes is a phospholipid bilayer. All biological membranes are made of the same basic structure. This is composed of molecules called phospholipids, which form a phospholipid bilayer. Phospholipids are fats. They're composed of two fatty acids tails and a phosphate head. The phosphate head are hydrophilic, whereas the fatty acid tails are hydrophobic, meaning that phospholipids are drawn to water at one end and drawn away from water at the other. When placed in water, the heads orientate themselves towards the water molecules and the tails away, meaning that the phospholipids will form a layer above the water if left. If phospholipid molecules are completely surrounded by water, they may form a bilayer. A phospholipid bilayer consists of two layers of phospholipids where the tails point inwards and the heads point outwards towards the water. One layer is a mirror image of the other. The phospholipid molecules are not bonded together. However, their amphiphilic nature gives the bilayer a degree of stability. Since the hydrophilic head cannot easily through the hydrophilic region move into the hydrophobic region created by the tails. The molecules can, however, move freely as the fluid in the plane of the bilayer. Small non-polar molecules can pass through the phospholipid bilayer since they can squeeze between the phospholipid molecules and are not repelled by the hydrophobic region. Water molecules can also move through the bilayer despite being polar. The fluid mosaic model states that membranes are composed of a phospholipid bilayer with various protein molecules floating around within it. The fluid part represents how some parts of the membrane can move around freely if they are not attached to other parts of the cell. The mosaic part illustrates the patchwork nature of the bilayer. Some proteins in the membrane are called intrinsic. This means that they completely span the bilayer. Others are called extrinsic, meaning that they are partly embedded in the bilayer. Carbohydrate polymers may attach to parts of the membrane, forming glycoproteins or glycolipids when attached to the phospholipid molecules, and glycoproteins when attached to proteins. Both glycoproteins and glycolipids can act as cell receptor sites. Hormones may bind to them, as may drugs, to initiate a response within the cell. Some intrinsic proteins are channel proteins. These are transport proteins that allow the movement of large molecules that would normally be too large to pass through the phospholipid bilayer. Other transport proteins are carrier proteins. These use energy in the form of ATP to actively move substances across the membrane. Enzymes and coenzymes may be attached to parts of the membrane in order to carry out metabolic reactions. Mitochondria have infolds of the membrane called cristae containing enzymes which are partly responsible for aerobic respiration. Steroid molecules or cholesterol gives the plasma membrane in some eukaryotic cells stability by reducing the fluidity and making the bilayer more complete. Hopefully, this video should have helped you complete your daily revision task. Please subscribe so you can keep up to date with other videos to help with your daily revision tasks. You can follow me on Facebook or follow me on Twitter to help you keep up to date.